I do this, Zoom people can see me. Hello on Zoom, hello in person. I'm very excited to introduce Brad Love as our MindCORE speaker today. I've been an admirer of Brad's work for a very long time. Somehow I've never actually met him. So I'm very excited that he's here to visit us and tell us about what his lab has been up to lately. So Brad got his bachelor's degree in cognitive and linguistic sciences at Brown, and then his PhD in cognitive psychology from Northwestern. He started as a faculty member in 1999 in the psychology department at the University of Texas, Austin. And then he moved to University College London in 2011, where he's still now a professor of cognitive and decision sciences. He's also a program leader in the human machine teams at the Alan Turing Institute. So Brad's lab studies how humans learn and make decisions. He uses modeling and behavioral experiments and neuroimaging experiments. And his work has revealed really foundational and mechanistic insights into a wide range of learning and decision-making domains, including how category learning unfolds over time across individual experiences, how attention guides our learning, how we learn in unsupervised settings, and how we navigate exploration, exploitation trade-offs, among many other things. So today his talk is on process models to understand mind and brain. Join me in welcoming Brad.
Yeah, thank you. It's a real pleasure to be here. I can't believe I haven't met you in person either, because I think just seeing so many talks and reading so many papers, I have like false memories or something, but I trust yours more than me. So it's a real pleasure to be speaking to a cognitive science group that does so much high quality research that's relevant to my personal interests. So like, what are my interests? Like, my interests are in how people learn, generalize, and make decisions, and with the computational and brain basis of those processes. And here's a joke that the philosophers and linguists would get to motivate my interests based on Quine's Gavagai thought experiment. So, imagine you see this rabbit hopping along and you hear Gavagai. Does it refer to the rabbit, a part of the rabbit, the hopping, a blade of grass, and so forth? So I'm interested in how people acquire and use concepts in these difficult circumstances. On this talk, there's going to be 4 parts. The first part is going to be basically an overview of some past model-based fMRI research we've done using cognitive models as a lens on brain function. In the second part of the talk, we're going to address a deficiency of those models. So cognitive models tend to be very abstract, but how do we get from there to something more neuron-like? So basically, how does the mind arise from the brain? How does we get symbols from neurons and so forth? And the third part of the talk, I want to broaden out to consider how we could use deep learning to work with more naturalistic stimuli and look at interactions across multiple brain regions. And the final part of the talk, I want to consider if we're going to use these more complex models, these deep learning models, we're going to need good ways of evaluating them, and I'm going to suggest a new way of doing that. Okay, so maybe just to back up, like, why does neuroscience even need models? So models play a number of useful roles. One is to specify theoretical ideas and make clear what we predict. You might be thinking, well, I know what my theory predicts, but historically, it hasn't always worked out that way. So just to use exemplar and prototype models as an example, exemplar models of category learning store every experience as a trace in memory. So if you're learning about birds, every example of a bird gets stored, whereas a prototype model would store them all in one node or cluster in memory, bring them all together. And so certain behavioral findings were thought to require prototype representation, but once these models are formalized, we realize that's not necessarily the case. And of course, one corollary of that is once you've made these models formal, then we could do basically model testing and see which one does the best with data. And that could be with behavior, but also with brains. We could look at brain data and see which one, do a kind of model decoding and see which models are most consistent with brain response. And finally, of course, once you have a model you like, it could assist in data analysis. So many people here do analyses in cognitive neuroscience, like representational similarity analysis, where they look at patterns predicted by their model and see where those same patterns of similarity persist in the brain. So you could use the model as a lens on what's going on with brain activity. And finally, there's this explosion, exponential growth in the scientific literature. And so models give us a way to compress or make sense of that vast literature. So it could be a useful conceptual tool to address our own limitations. So today I want to make more of a pitch for process models in particular. So what are process models? These are models that actually do the task that people do or the animal does in the lab. So they go through the steps that we think people are doing, and they're a mechanism because they have components that interact, and we can relate those components to brain activity. So there's many other types of models, but I'd like to just make a pitch for process models that they kind of put the model as the bridge between brain and behavior and potentially bring together, many subfields of cognitive science. So you could list more than this, but for today's talk, it's mostly psychology, neuroscience, and computer science. Okay, so let's jump in to this first part of the talk, which is mostly an overview of cognitive models as a window on brain function. So again, let me say what a cognitive model is. So it's a type of process model, but it's a very simple kind of model. It's very abstract. It's usually motivated by psychological principles. So these aren't models, with a lot like spiking neurons or ion channels. They're very, very, very simplified, but they're still formal models that are often verified in a number of behavioral studies, and we could use them as a lens on brain function. So let me show you a historic artifact from my graduate school days. This was made in XFIG on a sun spark station, and it shows an old clustering model. I'm going to just run through how it works because we're going to use this model and some model-based fMRI analysis. And so the idea is some stimulus comes in, it gets encoded along some features. And so in this case, the models learn to attend more to shape over, say, luminance. And there are these clusters. These are kind of the model's memory. Clusters are bundles of related experiences. You could think of them as like the model's concepts. But a category could have multiple clusters to it. And these clusters compete to characterize the stimulus, and one wins out that's most similar. And across these association weights, it conveys, a response. So in this lab task, let's just say category A or B. Let me say a little bit how these clusters form, because that's going to be relevant to later in the talk. Okay, so imagine we have this clustering model and it's learning. And so far it's learning about mammals and birds, and it's learned that mammals are big and they don't have wings, whereas birds are small and have wings. And we see this creature. So it's most similar to the mammal cluster because it's big and doesn't have wings. And so the model would predict it's a mammal. And so it would update its that cluster to reflect its newest member. Likewise, we see this creature. It's small, it has wings. It's most similar to this bird cluster. So it turns out we predict it's a bird, which it turns out it is. So we update. So far, this model is doing a kind of like pattern integration where related experiences are being collapsed together. It's really functioning like a prototype model. But where it differs is if you get a surprising example like this. So the model here would predict this is a bird. It's small, it has wings, but it turns out, bats are mammals. So surprising error that we have to like separate off. And this kind of recruits a new cluster, a new mammal cluster. And so now if we see, a similar bat creature, we'll update this mammal cluster and correctly predict it's a mammal. Okay, so that's really all you need to know about these models. And so that's a simple cognitive model. It does a great job accounting for a number of behavioral studies. Now we want to use it as a way to understand what's going on in the brain. And so we started thinking about that a long time ago and thinking about the kind of computations in this model. And at the time, there was some, but not very much imaging work. So it was mostly, patient studies and animal lesion studies. But this kind of computation seemed to relate to what goes on the hippocampus in coordination with medial prefrontal cortex of basically collapsing together relevant information and separating what needs to be separated. And so Tucker X and I put forth a theory like that in the modeled function of populations differing in function along the circuit. And we had a lot of like hostility from all quarters. So like cognitive people back then would just be like, well, what are you going to learn from neuroscience? There's nothing, it's not going to learn anything about the mind from the brain. It's kind of incredible, but that's what was the attitude then. But there's also like even more hostility from neuroscience. Like this model has nothing to do with the brain. Like where are the spiking neurons or the ion channels? What the heck's the clusters is also, idealized. So there wasn't really an appreciation of abstraction or thinking at multiple levels, which, there's still issues today, but it's improved a lot. But nevertheless, we persisted. And I used to work at Texas, as Anna said, and Allison Preston moved there, and with this good graduate student at the time, Tyler Davis, we did some basic model-based fMRI studies in which we fit this clustering model to behavior, and a learning study in which there were some items that followed a rule And then there are other items that were exceptions, like the bat, that violated the rule, and people had to learn through trial and error. And so we're just showing here some model regressors. So we put the model to behavior, and this is just a regressor related to what the model does at the time of decision. So it's sort of like a familiarity signal from the model across these learning trials. And then this other picture here is showing what happens at error correction. So at the portion of the trial where feedback's given, and you can see that this model is doing a good job, this clustering model, of characterizing what's going on in the hippocampus. So it's basically tracking the activity levels of the hippocampus, and that's not true of standard analysis, right? So if you just did a contrast like these exception items greater than rule following items, you pretty much get nothing. Or if you use the wrong model, so a model that we don't think is appropriate for this task. So in this case, this is an exemplar model that once again does not specifically differentiate surprising errors like these bat-like exception items. It doesn't really characterize what's going on in the hippocampus at the time of decision. So this is like an early win for verification, for this kind of approach. And we've gone on to do a number of studies like this. So Mike Mack, who's now associate professor at Toronto, we looked at changing the learning goal at the same set of stimuli. So in these clusters, it depends what you're trying to predict. So if you take the same set of stimuli and you create different category structures, you should get different clusterings, which implies different patterns of similarity across the items, different kind of structures. And so we did a representational similarity analysis to see if that same sort of pattern of differences holds in the hippocampus, and it does. So as the context of the learning task changes, the nature of the learning problem, so do the nature of the representations in the hippocampus. And with strong evidence, this was done in coordination with medial prefrontal cortex. Christiana Allenheim, who's now a data scientist at Google, and I were thinking, well, maybe these same sort of like attentional processes, figuring out what's relevant in different learning problems, affects not just the hippocampus and medial prefrontal cortex, but maybe representations, visual representations in the ventral visual stream. And so we thought maybe when there's more aspects of the stimulus that are relevant to the decision, in some sense, the dimensionality of the bold response in this case, using fMRI will be higher for those problems. And it turns out it is developing this method found support for that. And finally, in this review, we wanted to clarify what's going on in ventral medial prefrontal cortex. And we particularly thought it was relevant to determining relevancy, like, in a learning problem, which aspects of the stimulus did you attend to? And it looked like across the brain, ventral medial prefrontal cortex was unique in coming up with this goal relevant compression code of figuring out what's relevant to the problem to help facilitate learning. Okay, so more recently, we became ambitious and started thinking about, well, maybe there's just kind of one learning mechanism, the rule of the model. So there's a lot of interest these days, and the relationship between spatial and non-spatial concepts. And there's this idea that space might serve as like a, kind of a foundation for learning about more abstract domains. But we're kind of thinking, well, maybe there's just like a general learning mechanism. So if you feed in spatial information into this kind of clustering learning mechanism, you get spatial representations out. Where if you feed in something more conceptual, you get something that looks more like conceptual representations. So we tested out this idea and I tried to explain using the same learning mechanism, place and grid cells that are associated with a spatial domain. So let me explain what a grid cell is through this video of this rodent in this kind of typical experiment. So these are recordings from a cell, and every time the cell fires, there's a little white diamond plotted on the screen. And so if this were a place cell, it should only fire when the rodent passes a particular location. So that would be like what we would think of as like a cluster there. But you can see the receptive field for this cell is much more complex. It's forming this grid pattern. And so there's basically, if you look here, there's a six-fold symmetry emerging. So 1, 2, 3, 4, 5, 6. It's becoming more apparent. These are called grid cells. So how can these come about, and how can we explain how you would get cells like this out of such a simple learning mechanism like I've discussed? Okay, just like this. Really briefly, we think these responses are kind of unique to this type of experimental setup. So if you think about the structure of clusters and other domains, they're going to be very clumpy and sparse. So conceptual domains, there's only 2 dimensions shown here, but they can be very high dimensional. And you don't experience all combinations of all features. So for example, you don't come across elephants with wings and so forth. So, if you have a sort of a clustering of birds and mammals, most of the space won't be populated. In contrast, if you're like a rodent experiencing sort of this impoverished environment and just thoroughly exploring it, this is what the clustering will look like. It'll like completely tile the whole possible space. And so you kind of end up with this degenerative clustering that creates this 1, 2, 3, 4, 5, 6-fold symmetry in these clusters, which we think of as place cells. And so you could probably guess where the grid response is going to come from. But before that, just to, again, emphasize, this is the same learning mechanism, but in one domain, you end up with one kind of representation. The other domain, you end up with this other kind of representation, just applying the same simple learning mechanism. So again, where does the grid response come in? Well, something has to monitor the cluster match, basically an inverse error term. And so if the clusters are in this sort of grid-like pattern, which we think of as like the place cells, the clusters, then whatever monitors them is going to show a grid response. So that's what's being shown here. In contrast, if you're in a conceptual domain that's clumpy, whatever's monitoring these clusters, which we think of as like, concept cells or place cells, is not going to show a grid response. And so this is a really simple account, but it's done a good job of characterizing when and when not, and to what degree you see grid responses in various tasks. Okay, so just to summarize, we have this common learning mechanism here, and we applied it to different domains, and you get out different representations, but you're really using the same kind of approach. And so here, one prediction of this is the grid response should be relatively rare, and it should only come under these sort of degenerative clustering conditions, even in the spatial domain. And that's observed experimentally, like even in trapezoid environment, as opposed to circular or rectangular. To wrap up this cognitive modeling part of the talk, we reviewed how this surprise-driven clustering model does a good job of capturing human behavior, and it's hypothesized to rely on the hippocampus to build these clusters in coordination with ventral medial prefrontal cortex to establish like kind of attention, goal-directed attention or relevancy. And this has been confirmed in some model-based imaging studies, and we could extend this account to not just like category learning studies, but to spatial domains as well. And it's really kind of the same mechanism. But there's a big problem with these accounts. Yeah.
Maybe you don't want to go into this, but I actually did not understand how you get to the place cell to the grid cell. I got the,
oh, sure.
The place cells, you get this over, so you're kind of gonna get, categories or whatever, the place cell, so they're kind of distributed. Yeah, right, maybe the cell spaces or something. I don't know the place cell.
Okay, yeah, sure. So sorry, I covered so much material so quickly in that section. No, it's really, really simple. So the question is just clarifying, where does the grid cell response come from? So when the clustering, I think this part was clear, that if you have this sort of uniform sampling of this like rodent running around this, say, circular arena here, you're going to end up with a clustering that's just like a spear packing, basically. It's like if you throw a bunch of tennis balls in a hopper, they're going to form hexagonal patterns. And so that's what you see with the clustering here. Now, during learning, something needs to monitor that cluster match to direct learning. So basically what we're just saying is when you see a grid response, it's just a cell that's basically monitoring cluster match for a set of basically clusters, place cells, in the spatial domain. So here, if there's a cell basically monitoring all these cells, and every time one of these cells fires off, because these cells are in this like grid-like receptive field pattern, then whatever's monitoring them is going to show a grid response.
So the grid is actually there.
It's just whatever's monitoring the place cells. But if the place cells, sorry, the slide's hidden. If you were in a different environment that had, say, boundaries in it, or like more like a trapezoid type shape, then the clustering wouldn't look this hexagonal. And then whatever would monitor those cells would show a diminished grid response. Yeah. So that's the key. It's really that there's just not a lot of structure in this environment. So the clustering forms this sort of hexagonal pattern. And then whatever monitors that will show a grid response. And if people are probably thinking, well, what about Contino and from Barron's lab, the stretchy birds? Well, that's a case where once again, it's low dimensional, it's 2 dimensions, and all possible combinations have been tiled during learning. So this isn't spatial versus conceptual with grid response. It's just when you have sort of this degenerative clustering where you have all possible combinations of everything in low dimensions, that's when we predict you'll see a grid response. Yeah, I should have just given a talk on this. Okay, so that was like saying what's so great about this work. Now let me tell you why it's like terrible. Not terrible, but there's a huge, so this kind of goes back to the original criticism from neuroscientists about trying to say these cognitive models have something to do with the brain, because they're very high level and simplified, right? There's very few clusters, but there's many thousands of neurons. And so typically, what we do in others is just kind of wave our hands and just say, well, there's a many-to-one relationship between neurons and these higher level cognitive constructs like clusters. But how does that work? How is this coordination problem solved amongst those neurons to create these kind of like higher level data structures in the brain, almost like basically how does the brain make a symbol or anything higher level? So I want to consider that in this next section of the talk, how we could come up with a multi-level explanation. So not abandon the cognitive models, but how do we get from them to something a little bit more neuron-like? So I was taught as an undergraduate that there aren't grandmother cells. So this is like a fictitious cell that represents your grandmother. And if that cell were to die, you would lose all knowledge of your grandmother. So that's kind of ridiculous, but we do find Jennifer Aniston cells in the brain, or the field does. So these are cells that would selectively respond to Jennifer Aniston from different viewpoints and so forth. And so I like to point out this really isn't an explanation about how the brain represents like a concept or a symbol or something higher level. It's really an exciting finding that demands an explanation. So like, why do we see cells like this and how can they come about? Just to underscore how we don't really understand how like something like a symbol is implemented in the brain. If you think of an artifact we've made like a computer and we have like a sorting algorithm running, there's a symbols there, right? And we could go all the way down to like transistors or even physics, and we could understand how this symbol arises. Where in the brain, what we have is, we could have a cognitive model with something like clusters in it, and we could have a model based off fMRI, but these are really just neural correlates. We don't really have the same tight understanding about how the higher level arises from the lower level. Okay, so how does this coordination problem, how do we have all these neurons, can they give rise to like a cluster, or are these just incommensurate, dialogues? Okay, so the way we're going to try to crack this is taking a level of mechanisms approach, where at the highest level, you could think of this as just an input-output relationship, but then we can make a mechanism that implements that input-output relationship, like a cognitive model, like the clustering model. And so a mechanism has components that interact with each other. And importantly, a component could be decomposed into its own mechanism. So that's what we're going to do here. We're going to decompose the notion of clustering into elements that are more neuron-like. And make this intuitive, just think of a mechanism like a car. It has interacting components like an engine and a transmission. And we could take a component like the transmission and further decompose it into its own mechanism, take a part of that and decompose it and so forth. So we're just going to do that with the cognitive model, but for the clusters. To explain this coordination problem, how did all these neurons give rise to this like kind of virtual cluster? Because there aren't clusters when you open up your head. So how does that work? I started thinking about how like this might work in nature and other domains. And so if you think about in nature, there's things like flocks of birds, school of fish, and these are kind of like a cluster. So there's all these little elements that are coordinating with each other. And they're just doing things like matching each other's heading and so forth. So there's just simple local rules they follow that give rise to this kind of like higher order structure. And people have done this in artificial life simulations of flocks. So all these elements aren't controlled centrally, they're just like aligning headings with each other and following other simple local rules, but they're basically forming something like a cluster. So like maybe we could have some kind of neural flocking to create these cluster structures. Okay, so that's the basic idea we're going to pursue. And so the idea is we have this problem description and we have these algorithmic level cognitive models, and we want to sort of move more towards neural level. And so the idea with flocking is that instead of having one winning cluster now, we'll have thousands of neuron-like units that are winning, so they're more like neurons. And they'll update, when they update during learning, like the cartoon I showed you before, they'll all move towards their current member, right? So that's sort of a typical learning update. But there's going to be a second learning update that's going to make the coordination or flocking possible, that all these units are actually going to also move towards each other, okay? So all these highly activated neuron-like units will not just move towards the stimulus, but also towards each other. And that's enough to implement flocking or virtual clusters. And so how could this be implemented in the brain? Well, there's a couple of different ways. I mean, in internal recurrence in the hippocampus, where highly activated cells will align their receptive fields more. And there's also the so-called big loop recurrence that, you know, Darsh Kumar and Atit has discussed, where the output of the hippocampus could be recirculated through a neuronal cortex as an input. So effectively, the input could be recoded in terms of the response of the most activated units. So you could think of it just all the cells that are very highly active kind of moving to the mean of the receptive field. There's a lot of ways to do it, but the key is like these two updates towards the stimulus and then towards the highly activated units towards each other is going to turn out to be enough to solve this coordination problem. Just to show you a simulation of this, we revisited this cluster, this grid and place cell work, but now instead of with a few clusters, we could have thousands of these neuron-like units. And this is, let me just show you what it looks like when this rodent roams around. It looks like all of these units are disappearing. It's just they're not, they're just flocking on each other so tightly that they're forming basically like a symbol or a virtual cluster. And once again, we're going to get the six-fold symmetry, 1, 2, 3, 4, 5, 6. But now there actually aren't any cognitive constructs in this model, right? There aren't clusters. They're just these very small neuron-like units flocking with each other. So the clusters are kind of virtual. And doing this, we can still account for all the kinds of grid scores and so forth, and when you see grid response and when you don't. But we could also model cognitive domains. So here's a simulation of learning a simple category structure consisting of 2 categories. And there's 3 stimulus dimensions, but only one's relevant. This is a very simple learning problem called like a Shepard-Hobbling and Jenkins type 1 problem. So it only requires attending to 1 stimulus dimension. You can see the model forms 2 virtual flocks, 2 like neural flocks. So it's like 2 clusters. We ran this model on all of Shepard-Hobbling and Jenkins problems. These are category learning problems through trial and error, ranging from this really simple type 1 problem to this really irregularly structured type 6 problem, where basically every item needs to be memorized. So this is a set of problems that are hard to account for with cognitive models, the behavior. The clustering model I showed you does do that, it's a good job of it. Here's the behavior over here, and you don't have to really worry much about it. It's just that there's an error rate on the vertical axis, and the horizontal axis is the learning blocks. And the key finding is that the type 1 problem is the easiest to learn, then type 2, then 3, 4, and 5, then 6. And this neural flocking model could capture that same difficulty ordering that cognitive models themselves have trouble doing, even though there aren't really cognitive constructs in this model. Now, what's interesting too about the way it solves the problem is it solves it like the cognitive model it was based on, that it was decomposed from. So when the cognitive model, the simple clustering model, forms, say, 4 clusters, this more decomposed model with neural flocks will form 4 neural flocks. So it's doing it like qualitatively the same way, but it kind of with lower level constructs. And from those low level constructs, we can get additional insights, like why do you need so many neurons to code up these things? So if we introduce noise or like synaptic transmission failures or just kill some of these neurons off, everything works just fine when you have a number of neurons. It's just because, you know, a flock isn't defined by like, you know, 1 member of it. So it overcomes it quite easily. Okay, yeah.
Can you just elaborate on what determines the number of clusters? Like, you know,
Sure.
Find out two sides ago. I mean, why 6? Why not, you know, 6 times 12?
Yeah. No, it's a great question. So the question is like, what determines the number of clusters? So what's nice about it is it depends on surprising errors. So it depends on the learning problem. So to go back, if you have a really simple problem like the type 1 here, so there's basically like all the things that are lighter in one category, all the things that are dark in the other, the model doesn't make a lot of surprising errors. And so you end up with basically like a prototype model, 1 cluster per each category. Whereas if you're learning a very difficult problem like this type 6, there's basically no regularity. So you're constantly, you just end up memorizing every mistake. So it's a lot like the example I showed earlier with the bat. So if bats were birds, then you'd only have in that example 2 clusters. But Surprisingly, the world there has a little bit more complexity to it. And so that's why there was this sort of, back cluster coming up. So it's the same way with these artificial lab studies. It's just the patterns that, are surprising from the model's perspective lead to more clusters being recruited, which tends to slow down learning. And that seems to reflect what goes on with people. Yeah. So with the virtual clusters, it's really the same thing in terms of recruiting, but now we can't create neurons, at least not like this. And so it's just really cells are being sucked in to flock together to form these virtual clusters. So basically, yeah, we've basically taken this cognitive model and decomposed the notion of cluster, this cognitive construct, but we could go farther. So we could decompose, as far as you want to go in terms of what interests us. So we're going to do that a little bit further here. So there's one distinction within the hippocampus. There's many that are interesting, but here we're interested in this anterior versus posterior axis. So on the interior part of the hippocampus, you tend to have broader place fields, whereas posterior, they tend to be a bit narrower. And we see in our own work that if there's a simple learning problem with one with like few clusters where there's nice generalities you could exploit, they tend to be solved more in the interior part of the hippocampus. Whereas you see most of the fMRI result more posterior when there's exception items like these bat-like items, irregularities. And so what we did is we decomposed what we already decomposed from the cognitive model into two banks of neuron-like units, one interior, one posterior, where the anterior one had these really broad receptive fields. So they were tolerant to a lot of mismatches, whereas the posterior one was very tightly tuned. And we found that, the interior model would take over for the easy learning problems, consistent with seeing interior activations on those kind of learning problems, whereas the posterior bank of units would learn faster on the irregular problems that involve like a lot of kind of memorization and would take over on those learning problems, but collectively they gave the same aggregate behavior. Okay, so I just gave an account of how the brain implements symbols, clusters, and cognitive constructs based on this idea of neural flocking. And really the key idea there is that there's just two learning updates to solve this coordination problem. One with the elements moving their receptive fields towards the current stimulus, but also towards each other. And this can be implemented through various forms of recurrence in the hippocampus. And there's really no end in sight for this, right? So here we're focused on decomposing clusters, but of course, if someone cared about spiking neurons, you can keep going further and further. But what's nice is that we're not creating a patchwork of models. We're maintaining this link to the higher level account, this cognitive model the whole time, and just zooming in where the data or findings are of interest. Okay, so right now I'd like to broaden out briefly instead of diving down deeper and deeper to think about how to use ways we could use deep learning models to broaden out a bit. So another weakness of this work I've presented in the cognitive models is that it relied on these experimental defined features. So usually these binary distinctions like, is it a triangle or square? Is it small or large? Is it light or dark? And even with our own stimuli we use in our studies that look more naturalistic, they really just vary in these like stereotypical ways, like does it have the thick legs or the thin legs and so forth. But in the real world, like, life's more complicated. So here's this famous horse secretariat. And so if you ask people, what are the features of this horse? They'll say things like it has legs, but people also have legs, right? And tables have legs. So what do horses have? Horses have horses' legs, I guess, which seems a bit circular, but also it seems to suggest that the conceptual challenge of defining the features is the same as like defining the concept itself, the category itself. So how do we get out of this like quagmire? Well, it's not a cure-all, but one potential solution is working with deep learning models that could process naturalistic stimuli like photographs. So here's a classic deep learning model for object recognition that's trained on, say, like 1 million real-world images, and it's trained to assign them to one of 1,000 categories through end-to-end optimization using gradient descent. And so hopefully after training, we show a novel car to the model and it'll say car. And so now there's no experimental hand coding of features. So these are engineering models, but they end up being decent. They have their problems, but they're probably the best models of the ventral visual stream. So for example, this is a slide from Jim DiCarlo's lab. And so the canonical finding that's led to this deep learning revolution in computational neuroscience has been this early to early, late to late pattern of correspondence between model layers and stages along the ventral visual stream. And basically there's just like some kind of shared variance there where you could predict activity in say V1 from an early model layer, and you could predict in a later brain area like IT, you could predict it from a later model area. So I'll criticize this logic later of correspondence, but for now, let's just let it stand and say these end up being decent models of the ventral visual stream. In our own work, we're trying to move out of just object recognition to get, the goal is to get towards like looking at category learning without hand-coded stimuli. So here's some work towards that end, looking at hippocampal memory replay for memory consolidation. And so I won't go into detail just to show the kinds of questions one could ask with this sort of work. So we built this model of replay that worked with like regular photographic imagery, teaching it novel concepts. And we could ask questions like, what kind of information is replayed? And we found that if you use a very low res, almost dreamlike kind of stimulus for the model to replay, that works as well as actually a veridical stimulus, like a high resolution photograph. So that's kind of suggestive of what needs to be replayed. And we could ask questions like, where does replay occur? We find it's most beneficial in more advanced later layers, which makes sense when one thinks about semantic representation. We could ask, when is replay helpful? And we find it's most helpful for newly acquired and vulnerable information when people are learning categories. So these just showing that if this is, hopefully this gets paired with solid empirical work, it could be a way to ask questions at scale. Likewise, we got interested in how labeling affects semantic representation. So here, just a really quick example. We made a model of the ventral visual stream, but now we didn't use supervision. It was just unsupervised or self-supervised. And we trained this model to project images like real photographs, not cartoons like here, into this semantic layer, and then finally into this label, like using like the correct English word. And we found is we were able to explain some effects of labeling on like language on thought, effects of labeling on semantic representation. So basically very simple and intuitive stuff, like that when there's a common label, it tends to highlight get distortions towards common semantic properties. And when you have different labels for related things, it tends to differentiate them more. But it's kind of nice to do these at scale and be able to ask fine-grained questions. Perhaps most germane to today's talk, we've done some work in top-down attention. So how does basically how do your goals, how does the task reconfigure aspects of the visual system? And so here's an example you might have seen in interest psychology. If you haven't seen it before, it just looks like white and black dots. But if I tell you there's a Dalmatian in the photograph or to look for that, that's your target, then it usually pops out. And so we build in these kind of mechanisms from cognitive models into looking at how we would modulate activity along with the ventral visual stream to improve performance when the task context changes. Okay, so where this is all going, to get back on track, is again making models that don't require hand-coded representations for concept learning. So years ago, Olivia Guest, who's now at Donders, did a straightforward model which she took representations from these object recognition models and used them basically as the inputs into concept learning models. And she should have published it because subsequently people have done the same thing. It's rather high impact, but that's all right because we're on to something cooler. And so the idea here is we have these models, cognitive models, that are good models, at least we think they are, of aspects of the hippocampus and medial prefrontal cortex, but we're stuck with these hand-coded representations that we make up ourselves. So instead, it'd be nice if we could use these deep learning models, these models of the ventral visual stream, to kind of hand off to these models. And so rather than just having this perception, cognition handoff, we found that you could, if you reach a little bit further into the interior, a little lower in these deep learning models, that creates like a good representation for the clusters to learn from. But really what we want to do is ask bigger questions about how does like what the hippocampus and medial prefrontal cortex is prioritizing, how does it affect visual representations? So how do these areas integrate? So it doesn't seem like it could just be like, there's an input and then it goes to the cognitive bit. That perception and cognition are so separated. So we came up with this idea for an architecture, and this is really just work under development. We call it this like controller peripheral architecture, where you can have something that's a controller. say like the hippocampus. And the job of the peripheral, say like the parts of the ventral stream, are really just to serve the controller. So peripherals are like try to do is use the least resources possible, make the minimal effort while not disturbing processing in the controller. And so this is like kind of a new theory of attention in our case, because what we find here, like recapitulating some imaging results, is that when the hippocampal model, the cognitive model, says more dimensions are relevant, well, then you get sort of less sort of silencing of what's going on, higher dimensionality in the attention weights in the ventral stream. But you also get like, more precision for the stimulus attributes that are relevant for the higher level model, because at the higher level model, the controller doesn't care about something. It doesn't really have to be encoded by the ventral stream. So this is kind of an example we're pursuing of how you could look at interactions from more so-called cognitive and perceptual areas and how they might be organized in a quasi-hierarchical manner. Okay, so let me summarize this bit of the talk before getting into the last bit. So there's so much hype around deep learning, and a lot of people are justifiably skeptical, but for me, it's just really exciting to be able to move towards naturalistic stimuli and try to take on some of these conceptual issues. But I think in doing so, we should try to, move beyond these successes in object recognition and the ventral visual stream to get more of these learning and incorporate more control processes and top-down attention and so forth. Okay, so let me get the last part of the talk, because I think this is important. So if we're going to use these really complex models, we want to make sure, what do we mean when we say part of this model corresponds to part of the brain? How solid are our explanations and our logic surrounding them? So I mentioned this work from DiCarlo lab before, in which they use this encoding approach, in which they look at activity along a deep learning network, and they find that early layers, the activity there is good at explaining activity at early layers along early regions along the ventral visual stream. So there's basically a regression, right, from the model activity to explain the brain activity. And so this is an early, late to late pattern of correspondence, early model layers to early sort of brain regions, and along the ventral stream, late model layers to late regions. And there's a lot of other ways of looking at correspondence, but they all seem to come down to this shared variance idea. So for example, there's representational similarity analysis, but that's really looking at, again, like patterns of similarity at this model layer, and then looking at the similarity for the same items in the, say, the brain response, say the voxel response, if it's fMRI, and just looking at, do those similarity matrices match up? It's just looking at some notion of overall match. The same could be said of canonical correlation analysis or center kernel alignment, or like whatever approach one likes. It seems like they all just are based on some notion of correlation. And so just like, correlation doesn't imply causation, I don't think correlation implies correspondence. So I'm not quite sure, it might be worth re-examining this logic, given that much of the activity in a brain region might not be task relevant. It might not be, how do we know that's the variance we care about, the one that could actually drive behavior that is relevant. So we proposed a stricter test of correspondence. So if a brain region, so a model layer corresponds to a brain region, then you should be able to substitute that model layer with the brain activity and run the model computation forward, right? If two things are the same, one should be able to substitute for the other. So this is a kind of brain-machine interface type approach. Let me show you a picture to help illustrate this better. So up top here is just your typical deep learning model for object recognition. So we showed an ostrich, an image of an ostrich. Hopefully it says ostrich. Now let's say we thought this brain region corresponded to this model layer. So if so, instead of showing the image to the model, we'll show the image to the brain, take the activity from that brain region, use a simple linear kind of translation from brain space to model space, and then we should be able to drive the model forward along this red pathway, and the model should say ostrich, okay? So that's the idea of the substitution approach. If something brain region corresponds to model layer, we should be able to substitute in that one with the other and run the model forward to get the whole behavior output from the model. So that's what we're going to do. And the first data set we're going to consider is this bold 5000. We're going to see if we can still get this early to early, late to late pattern of correspondence between brain brain regions and model layers. So Bold 5000 showed participants a ton of images from a variety of categories. These are photographs. And so what I'm showing you here is the results for a later brain region, lateral occipital cortex. And what these bars are showing are when you stick in activity from lateral occipital cortex into early model layers on the left or late model layers on the right here. And so far this is consistent with the literature. The model, the deep learning model performs best. So this is the vertical axis, AUC, when you stick in this late brain activity into this later model layers. Okay, so far this is light delay. That's consistent with the literature. What happens if we look at an early visual area? So this is not what you'd expect based on existing analyses. So what we would expect, based on the current literature, is that the bars would be higher down here on the early layers. So if you stick in, say, V1, an early visual region, you think that would correspond to an early model layer. But it looks like you get the best performance from the deep learning model when you do the substitution to a later model layer. So this is suggesting that basically all the brain regions in this task look like late model layers. Likewise, you can look at things, other regions, and you find this sort of late effect. So that's just one data set. Let's look at another one. Again, this is a lot of photographs from 150 classes, 8 images per class. And we'll look at lateral occipital cortex again, which is a late brain region along the ventral visual stream. And what we're seeing here, this is consistent with the literature. When you stick in the brain activity into the deep learning model, try to drive it, you directly interface the two. It works best when you stick this brain activity into a late model layer. Okay, that's fine, but what about these earlier visual regions in V1, V2, V3, V4? So unfortunately for the zeitgeist of the field, we don't see early to early. These all also look like late, functionally like late model layers. You get the best performance when you stick the brain activity into a later model layer, and same for FFA and PPA. So maybe fMRI is just weird. Let's look at Jim DiCarlo Lab's data from 2 monkeys, looking at V4 and IT. So this is like multi-unit recording data. And these monkeys saw these strange images. And just as a sanity check, let's first, I'm going to reanalyze their data using our pipeline, not doing our approach, but doing theirs based on encoding models, based on shared variance. This is based on correlation, not substitution. And if you look at IT, which is a late brain region along the ventral visual stream, you can see it best correlates, this is not substitution, with later model layers. So that's late to late. Now, finally, when we do their analysis approach, we see V4, which is an early to intermediate. That's where it peaks in early to intermediate model layers. Okay, so it's just not that the field is analyzing their data wrong or we're doing it wrong. I think our results are showing something different. So now let's analyze the same data using our approach, the substitution approach. We had to make one allowance for their data because they didn't use standard ImageNet categories like cars and ostriches. They used these weird images. So we just made one minor change. We took our network and we just showed their weird images to the network. And we got an output across the 1,000 predefined classes. So like this 1,000-dimensional probability distribution. And we did the same for the brain interfaced thing. So here we'd show the image to the brain, stick in the activity into this model layer, and then we get output across the 1,000-dimension outputs of the deep learning model. And so the goal here was just to have these blue and red pathways give the same answer. And so now we used a measure of distance between the two, KL divergence. So now lower is just going to be better. Basically, does the model work the same when you showed an image versus drive it with brain activity? So it's really the same idea as before, just to deal with the strange images. So now lower is better. And when you look at IT, you can see once again, you get the best performance when you drive the late brain region drives the later model layers. But once again, with V4, which is the early to intermediate, using our substitution approach, again, it looks like a late model layer. So functionally, it looks like V4 looks late. So what's going on? So there's this whole zeitgeist, early, late to late. So what we think is going on is that there's long range recurrence going on from the later regions. And so after 60, 70 milliseconds, even if most of the variance in a brain region, like V1, V2, V4, is being driven by like stimulus properties, there's still this like signal, this almost like category signal of the class coming down, and it might not account for much of the variance, but it's really the task relevant variance, the variance that could drive behavior is there. And so that's what the substitution approach is picking up for. And if there's time in the question to answer, I'll show you some information flow Granger causality analyses that suggest that's what's going on. But this is not to say, just to reassure you, that you just don't always find everything goes to a late model layer. If you create a pixel level like image as opposed to brain data, you'd think that would best correspond to early model layer, and it does. This is like very high dimensional, like basically the image. And there, it'll map best this linear translation into earlier model layers. So it's not just that there's something intrinsic to dimensionality of the signal, the layers in the model or something. Yeah, so finally, just like to say that these models aren't without their value. They are picking up on some constraints from cognition by being trained on 1 million naturalistic images. So if you just apply like standard decoders, like linear SVM or something to the brain signals, you're basically a chance in decoding with the object classes, where you saw we did much better in driving these models towards correct answers based on brain activity. And one final bit is that it looks like these translations from brain to model space are very general. So everything I showed you was cross-validated with held out brain data and images. We could hold out entire classes. So for example, we could train this translation from brain to model space without using any ostriches. And if we show an ostrich for the first time, because it'll just have this general translation, it'll actually be very accurate and like this, what we call zero shot decoding or classification. So this seems very interesting and potentially useful for a brain machine interface, because it looks like it's a very general translation from models, from brains to models. Okay, so to summarize that section and wrap up, contrary to these approaches that dominate the literature based on shared variance, like encoding models, RSA, we don't find this early to early, late to late correspondence between brain regions and model layers. Using the substitution approach, it looks like everything after 60 milliseconds is like a late brain layer. And we think that's because of like this long range recurrence, basically carrying some task relevant information. Even if it doesn't dominate the variance, it's like can drive behavior. Okay, so to overview the talk and wrap up, I started out talking about using cognitive models as particularly these clustering models as a window on brain activity, and related them to what goes on in the hippocampus in terms and medial prefrontal cortex. So these models are very effective at accounting for behavior and bold response in those areas, but kind of like was a little bit hand wavy about how do these actually implement in neurons. And so we suggested that these cognitive constructs can be decomposed, taking this level of mechanism approach to something more neuron-like, and that we could just keep zooming in as we care, as we, whatever interests us, we care about spiking neurons, we could go in that direction. And so the whole point is to maintain this like higher level cognitive theory, but zoom in on what aspects of the brain are most interesting towards us. Another challenge for those cognitive models was dealing with real world naturalistic stimuli and not hand coding things, and deep learning provides a way there. And I tried to give some examples of where like large scale questions could be asked using these models, doing simulations with more naturalistic stimuli. But then one note of caution, these models are so complex that we need to be extra careful in how we evaluate them. And so I suggested that we should take this direct evaluation approach that I won't go through because I just went through it. But it basically, I think, is just a different logic of correspondence, that if two things correspond, one should substitute for the other. And so, it might not be that correlation implies correspondence. Okay, in terms of overall points, I tried to argue that process models are useful for computational neuroscience. So these are models that actually do the task. There certainly are tons of other kinds of models that are valuable, but I think these models, which have components that interact, provide good targets for localizing brain function. Again, allow us to zoom in as needed in the level of mechanisms approach. And I also try to emphasize there's been so many successes in object recognition. Maybe it's time to try to model other things at scale and embrace more rigorous model evaluation. I tried to acknowledge people throughout, but here's just a second shout out to some people, and just thank you. I know that was a lot, so let's cut you off, please.
Thank you for the presentation. I have a question about direct navigation approach, particularly early. So given the amount of affirmation before, I was wondering how how was the brain activation mapped onto the activation of the and given a number of different nodes at that earlier layers, could the problem performance earlier layers simply because there isn't a data express mapping problem?
Yeah, it's a really good question. I'll try to repeat some of it for the people online. But yeah, it was a question about how did we interface like these brain measures with the network? And so we actually did it two ways. One way was just complete, just basically like a regularized linear regression that we would just show a training set of images to the network, say the first layer of the network, and that would give some pattern there. And then we'd have the brain activity. And so that would just be like kind of the input to the regression. And then the model side would be the output. And so we just kind of train a kind of mimicry. So we tried to have the brain learn how to mimic the model's response to images. We also did it through backpropagation. It worked fine, but not as well, where you could imagine training the weights from the brain into the model layer to maximize the accuracy of the model. So both worked. We were worried that the backpropagation would have this sort of bias that you mentioned, that it could be this, even in modern networks, it could be like a vanishing gradient problem where it could create a bias to create, alignments with the later layer. And so you noted correctly that there's a higher dimensionality and number of units in the early layers in these deep neural networks versus the later layers. Fortunately, there's two things to say there. One, unlike an encoding model, we're going from brain to model instead of model to brain. So you could think of it as a bunch of separate regressions that predict those model units. So it's not necessarily bad if you're there's more things that you're trying to predict. It would be a problem if you're going in the other direction. The other thing to try to assuage that fear was that example I showed, that if we had something that was like more like a pixel type image, we were actually able to get correspondence from that to the early model layers. So it's just not that this can't work in practice. It's just that I think what's going on is that we have supporting analyses to show this, is that there just is a kind of a category signal after 60, 70 milliseconds in these early visual regions that is easier for the deep learning models, through this translation to pick up on than it is at the early layers. So I think just functionally, these regions are, through the logic of the substitution approach, more akin to advanced layers after 70 milliseconds. So this is like in multiple revisions right now. So one criticism maybe related to your question is like, well, if you don't have enough recording sites in these early visual regions, you're never going to be able to drive your model. Well, I think you can see that as a bug, but I think that's a feature, because if you're trying to say something corresponds to something, part of establishing correspondence, that requires like strong demands, require strong data, right? So like if you just record from one neuron, you can't say it corresponds to anything. And so if you have to record from more sites, well, I mean, that's tough. That's what it's going to take if you take this substitution logic. But I guess we just showed that it doesn't have to be that way. And it's also like, there's obviously like information in these cells because they're able to drive the model towards high level of accuracy, but only when you interface them with later model layers. And it's not just that, that's not trivial, because we put standard classifiers on those neurons. You can't pull out the classes, at least from the bold data. So I think there is something real going on, and it's just a little bit misleading to say that there's this early to early, late to late pattern correspondence after 70 milliseconds. That's probably too exhaustive of a response. Yeah, thanks. Yeah.
What if you guess right and guess wrong? I don't know, really really know how you the shape of the it's possible to make it kind of prove conclusion you want to make, you have to explore that state.
Yeah, no, definitely. So the question is like, are these deep learning models, they're so flexible, there's so many choices you can make in the architecture, number of units and so forth. How do you know you're getting it right? And I mean, surely they're not getting it right. And the model like I used wasn't even developed by neuroscientists. It's a very old, by deep learning standards, I think 2014 or something, VGG 16 model developed by engineers purely for engineering purposes. And there's very only recently in the last few years have even neuroscientists been trying to build in some constraints to the brain to develop their own versions of these models. Yeah, so I guess how people evaluate them in practice is basically like what I showed you. They look at shared variance. So like at MIT, you know, they have this brain score, which just looks at like how well, you know, basically an encoding approach, how much variance could you explain in the brain response from the models? And so it becomes a completely empirical question whether you've guessed correctly. Of course, you've never guessed correctly. Have you guessed like slightly better than someone else? And yeah, the theory is getting better on the deep learning side, but yeah, no, it is pretty still like, you know, a lot of just trying things out and seeing what works. Well, I guess maybe one nice feature of the work here is what we did is that we didn't make our own model. We took this popular model that does have a good brain score. So it does show an early to early, late to late pattern of correspondence. It's been around a while and we just used that. We didn't modify it. We just looked at just what we did learn is just this projection from brain data into the model to do the substitution thing. But yeah, all the issues you raise are valid for sure. Yeah.
So the place cell, good cell story. I'm wondering if you're saying that you call it a degenerate solution, and I'm wondering if that means you're saying that they don't have a function.
Yeah, this is really good. So like, I was just thinking of it degenerate, not like it's like amoral or something, but like it's like, you know, it's not like clustering, you want to pull out some structure in a domain and there's literally no structure. Like if you just have everything covered and sampled uniformly. So that's what I meant by degenerative. Like all the clustering model could do is like tile the space in this hexagonal sphere packing pattern. It's really funny talking about function. It's like interesting philosophically because it's like, it's kind of like, I always do this, everyone does it. It's kind of like teleological. So I see it as like the primary function, and maybe it's not even the right way to think, as like it's monitoring this, this cluster match, right? And that's all it's doing, it's helping to direct learning. But if that signal's there, it could be used by other things. So people that say like this grid code provides scaffolding, so that should only really work, I think, in like open fields and stuff. But if that's useful, and it seems like the brain would figure it out and use it for other things too. So yeah, a lot of it could be like, so I don't think it's representational per se, or just, I mean, it's not designed by anybody. So it's sort of like a tricky question when you start getting into it. But yeah, I guess just to say one concrete thing. Yeah.
But in this domain, it doesn't seem like it's obviously useful. You have to do something else.
Yeah. I mean, yeah, but if you get this sort of grid response, and I guess if they're like phase shifted, then you could start like implementing other people's models, but they are useful. But I'm actually not really, I guess a lot of these accounts, I know they're very popular, but it seems like, I think I just kind of like this like simplistic, you know, account. And it seems to do okay with the headline data. I mean, there's so many nuances in like place and grid cell remapping. And, but yeah, I think just getting the very basics of the degree of grid scores you see when you see grid response, when you don't. As a first approximation, this seems good. And like, you know, you suggested like, I'm saying it doesn't really have a function. I think the function is learning and like establishing these clusters. But just to repeat what I said, like maybe you could split that difference, because if that signal, even if I say it's for learning the clusters, then it could still be used for other things. Like maybe it could be used for path integration too in certain environments. Yeah. Yeah, please.
Could you talk a bit more about your display work that you presented? I'm wondering if your idea is if display contributes to blocking for it.
Yeah. So it's yeah, that's a good question. I never really thought about it related to the flocking. So what we want to do, there's all these popular approaches that are everyone, like neuroscientists really seem to like variational autoencoders, in which there's like this, basically there's this neural network and it kind of compresses to this central layer and then it comes out. But you can see it instead of going back up, kind of going back down, where you maybe have something like the hippocampus at the apex. So what we were thinking is, instead of doing that approach, let's just use these models of the ventral visual stream. And these representations could be in the hippocampus, but they could be like directing replay at different layers along the model. So instead of kind of that work with the autocoders, it's kind of stuck with this just doing replay kind of from one place. And so what was nice is we could look at sort of like this generative replay, but like kind of building, I also need more details about what we actually did, but we could look at it like, do you want to build sort of like kind of a representation of the category that's very low level, like in terms of the visual details, or more high level? And so we found that like the high level ones tended to work better in terms of promoting learning and generalization, which makes sense because it's not like you relearn your visual system, like when you're learning, you know, some new category, like why should the low level stuff, early stuff have to change much? Maybe it could be modulated, but why should it change very much? Yeah, and so what we did, the model was very simple. So we just, during learning, it would experience these examples, and we get these distributions of activity across various model layers. If we want to do replay in a particular model layer, we just built like kind of this multivariate Gaussian approximation of the activity patterns of that layer as these different stimuli were flying through the network. And then replay just sampled from them. So we just made these fictitious items from sampling from that multivariate Gaussian. And what was really surprising is that we have pretty low res, and they're pretty dreamlike. They don't really exactly look like, you know, a picture, but they were really good at like speeding learning rates and promoting generalization. I guess they captured enough of the statistical nature of the learning problem to like improve the network's performance. So yeah, I don't know, just like widely speculating, but maybe that's like why, you know, dreams or things could be so goofy or non-detailed. Yeah.
I guess since you have multiple different grid cells, phases or different scales, like there must be different sets of these phases that have their own that independently form United States sort of extend that to looking at how we do different sets of neurons that focus on different aspects of learning clusters.
Yeah, that's really, really interesting. I mean, I haven't thought, like the real expert in this is my collaborator, Rob Mock, who's the former postdoc research fellow at Cambridge now. Yeah, it's a really good idea. So yeah, the real power of the grid cells, like you're saying, and I think Anna's question was getting at this too, was you can't really kind of create almost like this Fourier type analysis or something like reconstruct, unless you have like things at different phases. So yeah, you're saying like, well, maybe you could have like various populations of place cells that are monitored by different things, different phases. Like, yeah, I haven't really, I should bring up with Rob to see if he could scare up any way that would be plausible or any reasonable evidence for it. But yeah, it's a really, really good point. I think just like everybody, we're probably a bit myopic. We're like, oh, well, this is how we think about it, and it works for our case. And we grab these headline findings, but it would be good. So like to try to see if there's a way to reconcile these other ideas with what we think, where they like you know, saying like they posit more of a function to these grid cells than we do. Where I don't really think of them as grid cells. I just think of them as like inverse cluster, kind of inverse error type cells. And I don't know, yeah. So that maybe that's why we haven't really thought about it, because we just didn't think they work that way. But yeah, we should be like better scientists and give that some thought. Some things. Yeah.
What extent do we have evidence that neurons, for example, look like flocking?
Yeah.
So I got the point that flocking mechanism capitulates the cognitive model, but then if it's
Yeah, no, it's funny, like yeah, hopefully I won't like crush his career with it, but I really, there's this Matt Brochard at who works in John Freeman's lab at Iowa is doing these like I think they're Utah array, like these unit recordings in rats doing these tasks. And so looking at concept learning tasks with rats, we've done the behavioral stuff before and some like dreads knockouts, but yeah, we're trying to actually see if we could find that. And one challenge is like, just the number of cells he records from is, it's good, but it's not like super high amounts, but yeah, it's exactly what we're trying to see if we could find that. And yeah, so I think you would need those kind of recordings to make it happen. So yeah, he's just showed me some results. It's not exactly related to your question, to do with the substitution approach, where it's like we really found like it's been really helpful actually trying to figure out the function, what's going on in just this rat. Where again, it's easier just to get like higher quality data in some sense, or different type of data than the fMRI provides. But yeah, so we're basically trying to do it, and I think like the jury's out, but we're trying to do it, and it doesn't look like it's hopeless. It looks like there's a chance. Oh yeah, but more the exact dynamics of like over time these cells would be highly active in regards to the situation, and then they would become like basically this subpopulation would just move closer and closer together and just coordinate across learning to create like a functional unit. So yeah, so it does, the Jennifer Aniston stuff is great because that's like what we predict should happen, that you should get cells like that. But I guess we would want to see is like someone learning about, I guess, I don't know, these animals aren't really learning about her, but they're learning about something that's creating that kind of coordination. Yeah, so this will just give us a chance to study it under controlled conditions where we could do our games as experimentalists and make our, our crazy stimuli that are patterned certain ways and well controlled, and see if we could, through learning, have these receptive fields change and flock like how we think. Yeah. Well, I don't know if that's it, but it's kind of exciting having so many people here in person. I know there's people online, but everyone should come in sometime because it's a really nice room. Yeah, thank you all. Great. Yeah, thank you.